Welcome, Nigerian medical and health science students, to your definitive Bio 221 revision session. The accuracy of clinical data analysis dictates patient outcomes, so mastering these rules is essential for your professional practice. We start with the foundational principle of biostatistics. The nature of your gathered data entirely dictates the mathematical test you must apply. You do not choose the test. The data chooses it for you. When you record continuous numerical measurements, like charting a patient's exact height in centimeters or their blood pressure in millimeters of mercury, the data usually follows a normal distribution. That allows you to use parametric tests, like the student t-test or ANOVA. But clinical medicine, microbiology, and epidemiology rarely present themselves in perfect continuous numbers. Scientists frequently deal with discrete, mutually exclusive categories. You aren't always measuring a gradient. Often, you are simply counting occurrences. A patient is either diseased or healthy. A cultured bacterial colony grows with either a smooth morphology or a rough one. Here is the unbreakable rule. When dealing with categorical frequency counts, parametric assumptions are violated. To extract meaning from this data, you must take an entirely different analytical path. This flowchart illustrates the biostatistical selection pathway. Notice how the decision tree isolates data types. The central non-parametric procedure used to handle categorical frequency data is the chi-square test. It funnels discrete counts away from parametric tests, pushing them directly into this methodology. Today, we will break down the two distinct operational pathways of this methodology, the goodness of fit test and test of independence. We will also dissect the critical examination traps designed to test your actual comprehension of these procedures. Correctly identifying the data type and selecting the appropriate non-parametric pathway is the mandatory first step in translating raw biological counts into evidence-based medicine. We begin with the goodness of fit test. This procedure evaluates whether a single observed sample distribution conforms to a specific theoretical distribution or an expected genetic ratio. This table provides the computational matrix for a bacterial colony experiment. A microbiologist has evaluated 250 total bacterial colonies, classifying them into three distinct types: smooth, rough, and mucoid. Theoretical genetic models predict that these phenotypes should appear in a specific ratio of 2 to 2 to 1. We need to verify if the raw counts match this theory. For this specific scenario, our null hypothesis states that there is no statistically significant difference between our observed bacterial counts and the expected 2 to 2 to 1 genetic theory. To calculate the expected frequencies, we add the ratio parts together to get a total of 5. For the smooth category, we multiply its fraction, 2/5, by the total 250 colonies, yielding an expected count of 100. The core chi-square equation measures deviation. You take the difference between the observed and expected counts, square that difference to remove negative values, and divide by the expected frequency to weight it. Then you sum those values across all categories. When we execute this calculation for all three bacterial groups and sum the results, we arrive at a final calculated chi-square test statistic of 0.75. To interpret this number, we need a critical threshold, which requires calculating the degrees of freedom. For a single variable goodness of fit test, the formula is simply the number of distinct categories minus 1, or k minus 1. Applying that to our experiment, we have three bacterial categories. 3 minus 1 gives us a degrees of freedom of exactly 2. At a standard 5% significance level with 2 degrees of freedom, our critical table value is 5.991. We compare our calculated statistic of 0.75 against this threshold. Because 0.75 is far less than 5.991, we fail to reject the null hypothesis. The math proves that the observed biological reality perfectly conforms to the predicted genetic theory. The second chi-square pathway is the test of independence. Instead of comparing one variable to a theory, this test evaluates whether two entirely distinct categorical variables are statistically associated within a single population. Consider this historical epidemiological survey. Researchers analyze 200 individuals to answer, is there a true, measurable association between smoking status and lung disease? This is a 2x2 contingency table detailing the survey results. The data is structured into intersecting rows representing smoking status and columns representing the presence or absence of lung disease. Here is where the calculation shifts. Because we are looking for an association between variables rather than testing a genetic theory, the expected frequencies cannot be derived from a simple baseline ratio. The expected cell frequency assumes total statistical independence. It is calculated by multiplying the marginal row total by the marginal column total and dividing that product by the grand total. To find the expected value for an individual who is both a smoker and has lung disease, we multiply the row total of 100 by the column total of 60, then divide by the grand total of 200. The expected count is 30. We run this specific calculation for all 4 cells, apply the standard chi-square deviation formula, and sum the components. The final calculated test statistic for this survey is 9.524. The degrees of freedom formula for a test of independence requires a different mechanical approach. It is calculated as the number of rows, -1, multiplied by the number of columns, -1. For our 2x2 table, that translates to 2 minus 1 multiplied by 2 minus 1. Our degrees of freedom is exactly 1. With 1 degree of freedom, the highly stringent critical table value is 3.841. We test our calculated statistic of 9.524 against this threshold. The calculated value vastly exceeds the critical value. Therefore, the null hypothesis is firmly rejected. By rejecting the assumption of independence, epidemiologists have demonstrated a statistically significant, non-random association between smoking and lung disease. The data proved the connection. Memorizing these formulas is only half the battle. Examiners actively design test questions containing procedural kill switches, hidden data conditions that instantly invalidate a standard chi-square calculation. This study guide outlines the most critical examination traps. The first major pitfall is the percentages distortion. Chi-square calculations must strictly utilize absolute, raw frequency counts. You can never plug percentages or proportions into the formula. If you use percentages, you strip away the total sample size. Chi-square relies entirely on sample size to establish statistical power. Converting counts to percentages directly distorts the degrees of freedom and yields an inaccurate test statistic. The second trap is the expected frequency threshold. If your math reveals that any single expected cell frequency is less than 5, the standard continuous chi-square distribution breaks down and fails to approximate the discrete probabilities. If you encounter an expected cell count under 5 in a 2x2 table, you cannot proceed with the standard test. You must substitute it with Yates' continuity correction or Fisher's exact test to get valid results. Internalizing these specific procedural boundaries separates rote memorization from genuine biostatistical competence. It ensures you never falsely interpret data in an examination or in a clinical setting. Success in Bio 221 requires organizing these isolated rules into a single comprehensive comparative framework so you can access them instantly under pressure. This revision matrix compares both methodologies. Goodness of fit evaluates single variables against theories, while the test of independence evaluates 2 variables for association. Their degrees of freedom diverge, yet both strictly require absolute raw counts and expected frequencies of 5 or greater. Applying these essential statistical rules directly marks the critical transition from theoretical classroom knowledge to professional, evidence-based clinical practice. This mathematical framework is the mechanism for evaluating modern public health data and interpreting complex genetic inheritance. Pause the video now, screenshot this final comparison matrix, and relentlessly test yourself against the active recall questions in your study material. Put in the repetitions, trust the data, and you will be fully prepared for your examination. The chi-square matrix provides the mathematical bridge that turns raw categorical counts into clear, statistically validated conclusions.